Okay, so this video is looking to see if age affects the proportion of slow and fast muscle fibres. So there are quite a few things that are really important to consider to get a reliable mean number of capillaries per muscle fibre. So that's what they were measuring. So what things were important that they did? So use the same muscle, and that's really important because the different muscles in the body have different proportions of slow and fast muscle fibres, so it wouldn't have been comparable if you'd have used different muscles. It was also important that they used several fields of view to make the results representative. It's also important that they chose the fields of view randomly to avoid bias. It's also important that they use the same species of animal. So they used hamsters for each one because different species would have different proportions of slow and fast muscle fibres. And another thing about reliability is the sample size. So yes, they have used several different hamsters, but it's actually quite a small sample size, so it's not really that representative. But it was a point that they used at least 8 animals in each group as a point for it. The next question is estimate how many slow muscle fibres were visible in the fields of view. Okay, so it says they counted 69 capillaries in the field of view. Okay, so, and it was for old slow muscle fibres. So there's 4.6, an average of 4.6 capillaries associated with each muscle fibre. So therefore, if there are 69 capillaries and a mean of 4.6 capillaries per muscle fibre, this would indicate that if you divide 69 by 4.6, there'd be 15 slow muscle fibres in the old hamsters in the field of view. Okay, question 3 is the actual number of slow muscle fibres wasn't 15, suggest a reason why. And the reason could be because there's standard deviations here. So this number could have been 3.6, 3.7 all the way to sort of 5.5, and that's just so 68% of the muscle fibres would have had between those two numbers, because plus or minus 1 standard deviation is 68% of the data. So there could be some muscle fibres with a lot less than that, and some muscle fibres with a lot more capillaries than that as well. So it's because you've got standard deviation, you know the spread around the mean. So there's a variation in the number of capillaries per muscle fibre. Question 4, why is it unethical? Because animals were killed. The next and last question, 5, evaluate the conclusion that age had a significant effect on the mean number of capillaries per fibre. So let's look at what we've got. So we can see that we have a significant difference between young and adult for both slow muscle fibres and fast muscle fibres. So when you factor in the standard deviations, we can see that they do not overlap between young and old, so young and adult for both slow and fast. Let's compare adult to old then. So as the mice gets even older, there's then not a significant difference for both slow and fast muscle fibres because the standard deviations overlap there, and the standard deviations will overlap here. So there's no significant difference for both slow and fast between adult and old. And the last thing we can say, if you compare young with old and young with old, we can see that there is not a significant difference with slow muscle fibres, because if you add 0.8 onto here, you get 4.2, and if you take 0.9 off from this, you would get 3.7. Therefore, they overlap. If you do between the young and old here, you get 4.8. If you add on the standard deviation, you get 6.2. So there's no overlap there. So there is a significant difference between young and old for fast. So significantly more fast fibres for in old than there is in young. However, in all of this, there is no statistical test, and so there's no statistical test, so we can't see if the difference is truly significant, and also because of the small sample size, it's not truly representative either. Do remember that you are expected to know so features of slow and fast muscle fibres as well as part of the spec, and how they can sort of make muscles adapted to do different jobs.